Hey everyone, and welcome to episode 599 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today we are excited to be rebroadcasting our interview with the fantastic Dr. Herbert Geller, originally released on January 21st of 2015. Now listeners, if you don't know Herb yet, he is a force in the field of neurobiology. Dr. Geller has conducted pioneering research in the area of neural recovery after brain and spinal cord injury. His lab identified a class of molecules in the extracellular matrix that impedes regrowth of neurons after injury, and they developed a novel therapeutic that could potentially help treat spinal cord injury. In addition, Herb is dedicated to mentorship and helping early career scientists, and in our interview, he shares some wonderful stories and insights. So sit back, relax, and get ready for this episode of People Behind the Science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to another episode of People Behind the Science. I am thrilled to be here today with Dr. Herbert Geller. So welcome to our show, Herbert. How are you?
Good. Glad to talk to you, Marie.
Wonderful. We are delighted to meet you today. Listeners, Herbert is a senior investigator in the Developmental Neurobiology section, as well as head of the Office of Education at the National Heart, Lung, and Blood Institute of the National Institutes of Health. He received his PhD in Biomedical Engineering from Case Western Reserve University and conducted postdoctoral research afterward at the University of Rochester School of Medicine and Dentistry. Herbert then served on the faculty at the Robert Wood Johnson Medical School for over 30 years before joining the NIH. Now, Herbert is a member of the Society for Neuroscience, as well as the American Association for the Advancement of Science. And Herbert is here with us today to tell us about his journey through life and science. So today, Herbert, we want to get to know you, both as a scientist, but also as a person. So can you take these first few minutes just to tell us what you like to do with your time when you're not doing science?
I like to stay active. So I think I come from a long history of carpenters in my family. And I think that was what originally drew me to engineering. And so a lot of my time, I like to try to build things or repair things. But at the same time, I believe in physical activity as a way to stay healthy. So I've been running. I've run some marathons, not as many as I used to. More recently, I've taken up swimming as a physical activity. And the most recent thing I've gotten into is actually growing some vegetables, being a vegetable gardener, because it's been good to grow some healthy food that we can eat at the same time as spending my time outdoors.
Well, it sounds like you have quite the exciting array of hobbies. I like it. I think definitely this idea of being active, getting out there, doing some running, doing some swimming, fantastic. And at carpentry, you say, what's the most recent project that you've worked on?
Well, actually, we have an old house. And I like to restore things in this old house. And it tends to be a lot of wood. And so, for example, my major project right now is restoring some of the older windows that a lot of people might throw them away and say, well, let's just buy new ones. But it turns out it's a lot of fun and challenging to restore them so that they work just like they were new and make the house just look great.
That sounds wonderful. Now, do you do a lot of fancy wood carving and stuff?
Not so much carving, but just mostly making things work well.
Gotcha. More functional. I like it. Well, Herbert, it's great to hear about how you spend your time when you're not at work. But now we're going to switch gears and talk about your scientific side. So can you tell us, how do you describe what you do to someone who might not be in science?
So I think what I do is almost easy to describe what we're interested in. The major scientific problem that we're involved in is why people do not recover from an injury to their central nervous system. So if you know somebody who's paralyzed from an accident, they're paralyzed. And currently there's no treatments we can offer these people. And our research is focused on trying to develop potential treatments for people with these kinds of injuries. Now, the major kind of response that we'd like to get is recovery of function. And recovery of function depends upon the ability of injured cells in the brain to regrow. And they don't in the adult. When you're a child, they're growing, but in an adult, after injury, they don't regrow. And you can easily imagine two reasons why they don't. One is that they can't, and the second is that something stops them. And our research is really focused on the second, the idea that there are stop signals in the brain that serve to prevent regeneration. And is there a way we can understand how these stop signals work? And is there something we can do to overcome them to potentially restore function to people with central nervous system injuries?
Certainly. Well, I think you've really taken us sort of to the ground level in an accessible way to get a little view into your research, which is phenomenal. Now, with all of these amazing questions they're asking in the laboratory, as well as the life that you're juggling outside the lab with carpentry, staying active, gardening, things like that, can you tell us today how you get it all done in 24 hours? I mean, I think that's one of those things that people always marvel at, people who are able to do it all, able to have this sense of balance. And I'm curious when I talk to scientists to hear if they have a favorite motivational quote or maybe a saying that really resonates with them and helps them get it done.
Well, there are a lot of favorite quotes that I have, and they really all apply to science. But one of them really comes from the fact that in addition to running and swimming, I also enjoy skiing. And in skiing, I had a ski instructor who basically said to me, if you're not falling, you're not trying hard enough. And I feel that's very important in science because you have to be trying to fail. Because in order to push the envelope, in order to try to push us into new frontiers and go where no one else has gone, you have to be prepared to fail. And so that's pretty much for me a motivational quote.
Absolutely.
And another one might be, don't waste your emotional energy on things you can't control. So there are a lot of things in our lives that we find that are annoyances and we have to put up with them. But if you really can't change it, it's better to just accept it and spend your energy being productive rather than try to tilt at windmills.
Absolutely. Well, Herbert, I think those are excellent motivational quotes. I really like this first one of almost pushing you to your limits, taking on maybe some of those risky projects, trying really hard in a way that it's okay if you fail because you know you were doing something worthwhile. And then certainly I like this idea of focusing your energy on the things that you have control of instead of fretting about all these things that you can't change. So great applications to science, I think, for both of those motivational quotes. Now, in addition to talking about some of the quotes that inspire people, I like to talk about some of the people who've inspired researchers. So can you think back on your journey through science and tell us about some of these people who inspired you or maybe served as role models?
Well, very early on in my scientific career, I was married to someone who also has a professional career. And in those days, it wasn't always so easy to envision how you'd have a personal life and also mixed it with a professional life. So there were two people, my PhD advisor and his wife were both faculty members at Case Western Reserve, and the Flemings were really our role models in the sense that they seemed like they could put it all together. They could have a professional life, they could have a personal life, and achieve. And so they were very strong motivators for us to pursue our careers as well as our marriage. And then later on, when I got my first faculty job, there was a senior faculty member named Ron Morris who really exemplified the person who would push the envelope and move into new fields periodically and then just create a new field. He didn't feel that it was important to sort of follow the crowd, but he really felt that it was essential to go and move into areas that were unexplored. And I think that was another very important lesson for me that I learned, that you can go into new areas and you can explore new areas. And again, it comes along with not being afraid to fail, but it also says this is the way we can move ahead in science.
Certainly. Well, it's wonderful to hear about these role models that you've had. And I think you touched on some really important points here. Just this idea of balance and really having people that you could watch and observe and sort of figure out how they do this in life. You know, balancing a career, balancing a personal life, balancing, you know, even hobbies outside of science is a challenge for a lot of people. So wonderful to hear that you had people in your life who you were able to emulate some of these qualities. And then certainly I love this idea of being adventurous in science and almost making your own path. So great to hear that you had someone able to teach you that lesson also along the way. Now, I'm always curious to talk to people, in addition about what are some of the people along their journey who inspired them. I'm always curious to know how they got interested in science in the first place. So, Herbert, can you think back now to the very beginning about when you first had that spark of curiosity, that interest in science?
Well, I was always interested in how things work. And so that's what made me and motivated me to go to school originally as an engineer. But life is full of surprises. And one of the surprises, and this mixes personal life and professional life, was that our family would spend time at a summer resort. And then one summer, there was a family that moved next door to us, and I actually liked the girl that was in this family.
A blossoming love story.
But she didn't like me so much. Her father did. And her father turned out to be working at Rockefeller University, at that time called Rockefeller Institute. And so it had got me a summer position at Rockefeller Institute the following summer while I was an undergraduate student. And during that time at Rockefeller, not only was I with the electronics lab, but the electronics lab dealt with helping scientists accomplish what they were out to do. And one of them was a scientist who was working on the nervous system, actually the visual system of the horseshoe crab, for which he got a Nobel Prize. And I kind of wandered into his lab and I said, gee, this is really interesting. This is something I could see myself doing. And so my first training after undergraduate school was to pick a school where I could do electrophysiology, which is what Keffer Hartline was doing. So that was my really first entry into science, but it was a major change in my career direction from being an engineer to being a life scientist. But it was all based on kind of a chance interaction. And I think that's, so much of a part of many people's lives, that you have to be open to these chance interactions that will open your horizons.
Absolutely. I think these chance interactions and these key people along the way often can completely change the direction of your career if it's just someone who's inspiring you, someone who's getting you excited about a particular field. So fascinating to hear about your experiences. Now, can you tell us a little bit about what happened next and how did you move from that point to really get to where you are today?
Well, in graduate school, I then did focus on electrophysiology. And again, here's another case where it was a chance interaction that I was just finishing up my PhD and was really thinking about what I wanted to do next. And I sort of knew the area that I wanted to learn. But by that time, I had decided I really enjoyed research so much that I could not see myself doing anything else but being a scientist with my own lab. And so if you want to be successful in that direction, you need to plan ahead. You need to think about what you need to learn and how you want to learn it. And I was thinking about what I wanted to learn and where I wanted to go. And then, in this case, another faculty member, a new junior faculty member at Case, came along and said, Herb, we know what you'd like to do, and I think you'd be successful if you go to work with this guy at Rochester who I know well, named Don Woodward. And I talked to Don, and it turned out, yes, we were great. It was just an absolute perfect fit. And I went to his lab as a postdoctoral fellow and continued my research on electrophysiology, perhaps in a slightly different direction, because, again, if you move into someone else's lab, you have to follow somewhat what they do. But then hone my skills as an electrophysiologist and then managed, again, by a chance interaction, to get a job interview at Rutgers, Robert Johnson Medical School, where I then moved. So I never had a situation, for example, where I, at least at that stage, where I was concerned about career. It sort of seemed to happen. But I think it happened because I was open to these experiences.
Absolutely. I think some of these serendipitous moments or connections, I think a lot of scientists have been talking about. And I think it's interesting to hear about how people maybe move from one institution to the next, or how they go from position to position, or even from different aspects of a field because of the people that they're meeting, the connections that they're making. So fascinating to hear about your journey. Now, can you tell us, you spent quite a bit of time there at the Robert Wood Johnson Medical School. What then brought you to the NIH where you are today?
Well, in fact, as I mentioned, I had a wife. And as it turned out, she was recruited to come to NIH. And after a while, we decided that we didn't really like living apart, with me living in New Jersey and Nancy living in Washington, D.C., and seeing each other weekends. So I began a job search to see if I could find a position which was suitable for me at NIH. And luckily, there was one which, as you mentioned, it combines both my ability to do some science, which I find really exciting, but also the ability to mentor the next generation of scientists as part of my job as head of the Office of Education. So here again, it was both a family and a professional reason for me to make this move to Washington, to NIH.
Well, that is very exciting. I'm glad you found that position. That is such a great fit for you in terms of being able to do science, as well as having interactions with students as part of this head of the Office of Education. So can you tell us a little bit more about your responsibilities there?
Well, in the Office of Education, what we're interested in doing is making sure that people who don't have their final job, so in other words, everybody who isn't tenured, so that includes everybody from our summer students, graduate students, postdoctoral fellows, and even junior faculty, know what it takes to get to the next level. But it also implies that we help them figure out what the next level is. So for faculty, it's not so hard to know what the next level is. They like to get tenure. But for graduate students and postdoctoral fellows, there are lots of career choices these days. And so what my job in the Office of Education tries to do is help people figure out what they like to do best and how to use their skills in a career where they'll be happy.
Very interesting. Well, definitely an important role, particularly, I know there's a lot of graduate students out there who are not sure if science is for them, or they're starting to second guess because of the funding situation, different obstacles that are standing in their way. So very exciting to hear that you're able to help them along their journey to figure out what is the best fit for them. Now, we've talked a little bit about your research in the beginning in terms of what you're studying. Can you dive into a little more detail on one of these projects that you're working on right now that you are so excited about, Herbert?
Okay, so the project that we're really very excited about is that I mentioned at the beginning of the talk, we work on molecules that essentially provide stop signals for neurons growing. And one of those molecules that really has a prominent role after injury to the spinal cord and brain is a family of molecules called chondroitin sulfate proteoglycans. And that's a long word, but it really has three components. Chondroitin sulfate is a family of sugar molecules. And proteoglycan means that these sugar molecules decorate proteins. And our research is involved in showing that in the signaling from chondroitin sulfate proteoglycans to stop neurons growing, that it's actually the sugar that does the signaling. So in that sense, we're sort of neuroglycobiologists. And so the question is, how do chondroitin sulfate proteoglycans signal to neurons? And what we've shown is, in fact, that the sulfate, and it's chondroitin sulfate because it's chondroitin sugars that are decorated by sulfate groups. And it's the positions of these sulfate groups that actually seem to matter. It's sort of like the sulfate groups on a chondroitin chain are just like the little positions on a key that allow it to fit in with the lock. And what we've discovered is that there's a particular enzyme called aryl sulfatase B, which removes just one of those sulfate groups from a very specific position all the way at the end of the chain. And just by taking off one of those sulfate groups, we can eliminate the biological activity of these chondroitin sulfate proteoglycans. And then when neurons confront them, they will grow. So we think that this enzyme, aryl sulfatase B, has a real potential for treating spinal cord injury in people. And so one of the things we're trying to push is to do the appropriate series of experiments that actually test that hypothesis and evaluates the ability of this enzyme to actually make things better.
Well, that is a phenomenal research project. Now, do you think this is sort of the fix-all solution, or do you think it's going to end up being a combination of things that you administer that will be the successful treatment?
There's no question that it's going to be a part of a combination. Spinal cord injury not only has these stop signals, but as I mentioned, the real fact that adult neurons don't grow that well. And so what we need to do is also have a signal inside the neuron that says go.
Right.
I think we're going to have to use a combination of enzymes that will provide both a go signal to make neurons grow and eliminate the stop signal or make neurons overcome the stop signals. Unfortunately, it's not so simple. It's not like a magic bullet.
Right.
Brain injury is a very complicated situation for which a combination is essentially going to be the solution.
Certainly. Well, it is exciting to hear that you may have found one of these key pieces to the, sort of cocktail that will be delivered to help improve the condition of people with spinal cord injuries. So that is phenomenal work. And I think so far, Herbert, we've hit a lot of these high points along your career and your journey as a scientist and talked about some of the exciting work that you're doing in the lab. We haven't yet touched on some of the struggles, some of the difficult times in science. And I think every scientist goes through tough times as well. And I think it's important to talk about these so people have a sense of what it's like to be a scientist. And I think the successes don't always happen as frequently as we'd like. And we run into some stumbling blocks, some troubles along the way. So can you take us back to a point in your own career where you were struggling with something, or maybe you experienced a major failure, and tell us what happened and what are some of these steps that you took to get yourself through that difficult time?
I think all scientists right now are struggling with the situation of grant funding. And of course, when I was an academic, not every grant application I wrote was funded. But I think that's unfortunately a very common situation in our times. But let me tell you about a situation where I really encountered a difficulty, and ultimately the difficulty turned out to be the most beneficial thing I've ever happened to me.
Sure.
I had originally trained as an electrophysiologist. And as I was about 7 or 8 years into my career in New Jersey, I was due for a sabbatical. And I had decided that I really needed to learn this technique. And in those days, it was very hot. Nowadays, it's very routine, of using monoclonal antibodies to identify different cell types. And so I picked a lab in London that I thought they had published a lot with monoclonal antibodies, and they were really experts. And I thought, if I went to this lab, I really would have a great shot at incorporating this technology into my research.
You could learn the secrets, of course, yes.
So I got there, and much to my surprise, they had moved on. And they no longer were interested in monoclonal antibodies.
Really?
So I got there. I had a year in London to do something. I had obtained fellowships for this year. And here I was thinking, what am I going to do? It was like, I can't just do nothing. So after sitting down with the head of the lab, Marty Raff, who's just absolutely a wonderful scientist, he threw out a bunch of projects. And as it turned out, I picked the one that I knew the least about because I figured, again, I wasn't afraid of failing. I just thought, well, I better take a chance. And this seemed really like pushing the envelope. And this was the best thing that ever happened to me because instead of retaining my interest in electrophysiology, which is how neurons work, I ended up learning molecular biology and cell biology and changing the entire focus of my research from how the neuron behaved in a physiological sense to the other major cell type of the brain, which are the glial cells. And it turns out that it's the glial cells that make these chondroitin sulfate proteoglycans that inhibit neuronal growth. And if I hadn't sort of been disappointed at not being able to do monoclonal antibodies, I probably never would have opened up this whole other window into this area of research, which has really been what I've been doing for the last 30 years. And it was really devastating to find out that they didn't do what I thought we'd
Right. Things you'd been looking forward to.
I had built up this whole idea and experiments, but then by the end of the year, I had created a glial cell line, which was the first ever, and learned a whole lot of new biology, which has enabled me to really blossom in this other area, which I never would have done had that initial failure happened.
Absolutely. Well, this sounds like one of those situations where it's almost a happy accident that really had a big impact on your career. Like you said, if you had just gone there, maybe learned a few things about monoclonal antibodies, and then come back and resumed your research as you had been doing it, you might never have gotten to where you are today answering some of these questions that you're answering. So phenomenal to hear about some of these turning points, some of these challenges that scientists went through that ended up having a happy ending. So we don't just want to talk about the tough times. I love talking about these success stories. So can you tell us next, Herbert, about one of these successes, maybe that's happened recently, that's really meant a lot to you?
Well, I think, as I mentioned, we've recently discovered how this enzyme called aryl sulfatase B might actually be useful in the treatment of spinal cord injury. And that's been immensely gratifying. And one of the real exciting things for me is, number one, is that aryl sulfatase B actually happens to be an approved drug for use in people because, unfortunately, there are a bunch of people who are born with mutations in the gene that codes for aryl sulfatase B. And so they have a disease that's treated by this enzyme. And so the enzyme is actually able to go immediately into people if we can show enough preliminary data in animals. And so what I've found is that there are some companies out there that are interested in taking those next steps because they might see a path to get this to the clinic. And I feel that's really one of our success stories lately. Now, obviously, it's not there yet, but in terms of the way science goes, you're never there.
Right. So can you tell us, I mean, this is a really exciting discovery. When you first saw the data that maybe first demonstrated inklings that this enzyme was going to be potentially effective, what were you thinking?
We were overjoyed. We really didn't know that this was going to be the result because in order to show that it had its effect, we had spent about a year developing some new biochemical techniques to actually measure how this enzyme might work. And so we had put a whole lot of energy into the story well before we got the result. And so it was extremely gratifying, not only to me, but to the people actually who did the pipetting, who were the postdocs and the people in the lab, who we were all quite excited to see kind of when neurons started to grow in our culture system, which is the test we do. We were incredibly gratifying to see that happen.
Absolutely. I think everyone in the lab is so invested at that point that it's really exciting to get those results and sort of see what you were hoping to in science, which I think doesn't happen nearly enough. Oftentimes you're disappointed by the results. So
I think we have to temper that. But yes, we were happy to see these results. But I think the other thing I tell people is, don't get emotionally involved in your data. As a scientist, I think it's always important to be dispassionate because if you get too emotionally involved, if I said we must have this work, then it could lead to the kinds of unfortunate situations where people overinterpret or kind of force their data to answer the question in a way that they want it to be answered.
You start seeing patterns that aren't there because you're so hopeful. Well, Herb, it's great to hear about one of these success stories coming from the lab, and I think a really exciting direction for your research. So wonderful to hear about that. And I know scientists keep so busy in terms of the work that they're doing, the research that they're investing in. And I think it doesn't always leave a lot of free time for scientists to maybe relax, enjoy some of their hobbies, and enjoy a good book. But I've gotten some incredible book recommendations from scientists who we've had on our show. So can you tell us today, Herb, about a book that you've read and really enjoyed that you'd recommend for our listeners?
I read as much as I possibly can, but it's hard to say one book. I think a book early on that was essential for my development was Catch-22 by Joseph Heller.
Great book. Yeah.
A great book that I think everybody should read because the lesson you learn about how these people interacted with each other under the situations of the war was unbelievable. But really, I tend to read authors rather than one book by any author. So, for example, the authors I really favor are, I think, Primo Levi is an outstanding author who talks about his, unfortunately, is no longer with us, but he talked about his situation in Europe. Robertson Davis has written a series of trilogies which are very magical and are a good read for anybody. And more recently, I sort of a more contemporary author would be Jonathan Safran Foer, and almost anything he writes is a good read.
Well, those are outstanding book recommendations and author recommendations as well. So I'll put those out on our website for our listeners to check out if they are looking for a good book. I know I always like getting recommendations from people. It's interesting to see what other people are reading, what they have on their bookshelf, and great to hear that you're able to dedicate a bit of time to reading. And I know one of the times that I tend to get the most reading in is when I'm traveling, when I'm sitting on an airplane. And I know scientists do actually get quite a few opportunities to travel, to go see the world, to share their research. So Herb, can you think back now on all of these places that you've traveled for science? And can you tell us about your favorite place that you visited?
Well, I mean, one of the wonderful things about science is you get to travel the world. So science has taken me all over. But I tend to like, rather than being sort of going to a meeting, spending 3 or 4 days in a city, to actually spend time in a place. So the two places that I really have spent time in that I really enjoyed, the first was, I mentioned the sabbatical time I spent in London, which was sort of a magical over a year in London where I was able to do both science and enjoy the immense amount of culture that takes place there. And so I put London right at the top of the list of places that I certainly enjoy going back to in all of Britain. And then the second place where I was a visiting faculty member, which I really enjoyed because it was so different, is Japan. And being in Japan and doing research in Japan where the culture's a very different culture. And so it allowed me to really immerse myself in a very different culture and learn how people behave. And it's really, I think, a wonderful experience of being a scientist to experience all these different cultures. And I've been very lucky to be able to do it, not just in a short-term visit, but over an extended period of time.
Certainly. Well, I think you brought up a really great point there. I think scientists do get to travel a lot of places for these short stints, these conferences, things like that. And you're often exhausted because you spend all day at the conference and you try and maybe pack in some activities in the evening. And, maybe at the end of 5 days, you're just ready to sleep. So it doesn't quite feel like you've gotten to enjoy the culture, to relax, to kind of immerse yourself in it. So wonderful to hear that you've had these opportunities to have a more prolonged stay in different places. Now, is there a particular place or a particular site in either London or Japan or both that really stuck out to you and maybe was your favorite place to spend your time while you were there?
Well, in London, there's just so many different places to go. And for me, going to the theater in London was one of the high points of our experience, is that I could spend my time in the lab during the day and we were able to go to the theater at night. And the theater in London is perhaps unequaled anywhere. I mean, New York probably comes close, but in those times, the theater was just exceptional. And so it wasn't any one place, but the idea of having this culture available to us. And in Japan, I think it was just experiencing the streets, experiencing how different it is in Japan, in the Western world, and experiencing how people interact with each other. Obviously, downtown Tokyo is unbelievable. The Tsukuji district and shopping in Tokyo was just exceptional. And so I think that it's, rather than any one place for me, it's experiencing the entire culture that makes the experience memorable.
Certainly. Well, I think that's one of the exciting things about visiting cities outside of where you're from or outside of where you're living, is just being able to get a sample of the culture. So I love that you're describing going out, being able to enjoy the arts, being able to enjoy theater in London, and even just being able to take it all in, walking around on the streets of Tokyo. These both sound like phenomenal experiences. So I like to chat about these different travel experiences that scientists have had. And I think when you look back over your career, not only the travel that sticks out, it's some of the people that you've met along your journey as a scientist. And I think a lot of scientists that I've met and worked with are just amazing, creative, and oftentimes very funny people with incredible senses of humor. And I think when you get so many brilliant people working close together in a laboratory, you get some quirky traditions that spring up and maybe just some funny memories that you shared together. So can you tell us today, Herbert, about one of these traditions that you've experienced, or maybe a funny memory that you have from science?
That's a hard one. I mean, because our research has sort of evolved over the years, it's hard for us to have sort of experienced traditions. I think every lab has its own little culture, but I think one of the things that I try to do, one of my thoughts is that a hungry researcher is not a happy researcher.
Right.
So the tradition that I try to bring forward, and maybe it's perhaps my heritage too, is that I think food is a very important part of helping smoothing communication. I try to make sure that everybody has some munchies around when we have our meetings so that if things get tense, there's some chocolate around that can sort of soothe people.
Improve the mood, yeah. I like that. So do you have a snack of choice that you like to have around during lab meetings or during one of these conferences or things like that you're sitting around with your lab mates?
Yes. Food really and snack, they help communication.
Absolutely. So what's your favorite, Herb, if you had to pick?
Well, I have this huge jar of jelly beans.
Really?
And I keep it in my office because it actually serves as an attractor. Everybody knows that there's a jar of jelly beans that they could hit up.
They want to come visit you then. So
I encourage them to visit.
Absolutely. I think jelly beans are dangerous too because you never feel like you're eating a lot of them because they're so small. But well, I think that is a great tradition. And I think definitely something I think a lot of people will agree. It's wonderful to have snacks around, food around, something to ease the tension and relieve stress a little bit if you're able to enjoy a little snack there. So great tradition to have in your lab. And I think, while you're snacking, while you're talking to the people in the lab, you often come up with these big research questions. And it's fun to talk about all these hypothetical questions, things that if you had anything, you could answer. So I'm going to ask you about that today, Herbert. Can you tell us, if we gave you everything, if things like funding and staff and technology and feasibility all were not an issue, what is one big research question that you are just dying to know the answer to?
Well, as a neuroscientist who came out of engineering, I tend to focus on sort of mechanistic things like neuronal growth and how can we make neurons grow. But I think if you really are on a more global sense, if we think about how the brain works, the brain isn't only there to move your arms and legs. It's how we generate thoughts and how we create memories. And how these are made and stored in our brain is, for me, a great unexplored question that I certainly, if I had enough time and energy and maybe another life, I'd like to think about how the machinery of the brain, because even though we have a bunch of connections in the brain from one cell to another, these properties of memory and thinking and ideation are essentially emergent properties that are not simply functional based on these connections. So there's something else going on that I don't think we understand, and I'd love to understand how we create these situations out of a sort of a biophysical brain.
Absolutely. I love this big question. I'm a neuroscientist myself, so I get excited about these types of questions. But just the thought about translating from basically the mechanisms, the nitty-gritty details of the brain to, what gets that to the point where we're having our own experiences, having our own motivations. So these are fantastic questions to ponder with your lab mates. And I think you're absolutely right. We need a couple extra lifetimes and perhaps some advanced technology, different things that we could do to start investigating these big questions. But definitely wonderful to think about now. Now, I know, Herb, when we talk about the career of a scientist, I think when you look back, we've talked about some of these mentors that you've had over the years. And I think scientists get a lot of great advice from a variety of different sources throughout their lives, throughout their careers. And I like to ask people about what they would consider just the best piece of advice they've ever received. So can you share that with us today?
Well, I think the best piece of advice I ever received is don't follow the crowd. I mean, whatever is trendy today in science is going to be passé in 5 years or 10 years. So if you try to follow whatever the trend of the moment is, and I'm not going to elaborate them because I will alienate all the people who have done that and continue to do that, is that you really need to follow your own path. You need to identify areas where there are important questions which are essentially unexplored, rather than trying to go into an area which is already well populated. Because one of the things that can happen in that situation is there's just a lot of data, a lot of it's going to be wrong, and you're going to spend most of your time trying to correct the mistakes rather than move ahead. So it's much more rewarding in science to essentially move into your own area and set the trend rather than be a follower.
Excellent advice to really make your own path and make your own space within science rather than trying to always keep up with what everyone else is doing and take on all these maybe new techniques that don't necessarily fit with your research questions or your research portfolio as well. So great advice for scientists out there. Now, Herbert, is there any other last piece of advice or maybe a last piece of inspiration that you want to share with our listeners now, coming straight from you personally?
Well, I think for me, having been a scientist for quite a while, I look back and I think it's just been an immensely rewarding career. You're never done. When you get to what you think is the top of the mountain, well, then you see the next peak.
Right.
So there's always a challenge ahead. And so I think the idea that, yes, you make progress, but you always have something else to go. So obviously it's incredibly rewarding to make the discoveries, but you also have to be ready to be frustrated because you're never there. So whenever you have a failure, you have to just be ready to pick yourself up and move forward, not dwell on it, but just know that there's something positive ahead. And I think that's the most rewarding thing, I think, for science is the challenge of discovery balanced by the frustration of failure.
Very well said, Herbert. I think that definitely captures the excitement of science, the spirit of science. And I think you're absolutely right. You have these moments where you're so close, you get there, and then you realize you're not done. You know, there's always more to do. There's always more questions that come out of every experiment. There's always new directions that you could go and new ground to cover. So wonderfully said, and great note of inspiration to end on here. So we've had some excellent conversations today. Can you tell our listeners, Herbert, how they might be able to reach out to learn a little bit more about you or your research?
I'm always happy to talk to people. We have an email address. Obviously, email is very valuable communication. Geller@helix.nih.gov works, and anybody certainly who wants to learn more about what we do, I think if you just Google me, you'll find our website, which talks about our research and what we're trying to accomplish.
Wonderful. Well, listeners, definitely check out their website, see what else they're working on, and certainly feel free to reach out if you have any particular questions. And Herb, it was wonderful to chat with you today. And thank you so much for joining us and sharing a piece of your story.
Great to talk to you, Marie.
This was a pleasure for me as well. And thank you to our listeners for joining us. We'll see you next time on another episode of People Behind the Science. Your voyage to the lives of today's exceptional scientists has just begun. You can find everything we talked about today, including our guests' favorite books, biographies, photos, and more, when you visit us at www.peoplebehindthescience.com. I look forward to seeing you next time on People Behind the Science.